Well, hello, seaweed true believers. This is Courtney Stepien, and I'm here today to talk to you about how seaweeds and other marine plants like surf grasses use carbon in the oceans. Now, before we get started, you might be wondering why do we care about this? This is a good question. So, when a lot of people think about climate change, we hear a lot about CO2 in the atmosphere, global warming, but a lot of things, the flip side of this is the oceans act as big sponges, big sinks that just suck up all that extra carbon dioxide. And that's actually changing the ocean chemistry, how much carbon is available to seaweeds, what type of carbon is available to them, and these can have some implications for ocean communities and ecosystems down the line. So let's kind of dive into what all that means. So when I say that there's different kinds of carbon in the ocean, it's a little more complicated. Photosynthesizing underwater is a little more complicated than what you do on land. So any plant in your garden, CO2, carbon dioxide from the atmosphere, it's floating around, it diffuses into the leaf, or is more actively concentrated by the plant, and then photosynthesis happens, and then you're golden. So underwater, it turns out that CO2 is actually super rare underwater. If you take a look at the bottom part of this slide, it's only really 1% of the dissolved inorganic carbon. There's really not a whole lot going on. But one big player of carbon, big carbon source underwater is bicarbonate, this HCO3 minus. Now, in order to tease out which seaweeds are using carbon dioxide only and which seaweeds can also use bicarbonate, this is an active process. It takes energy, takes some skills as a seaweed to use bicarbonate. So how do we figure out which kinds of carbon seaweeds are using so we can understand how they might respond to climate change in the future and changing carbon dynamics underwater? So if we take a look at the upper left plot up here, it turns out that bicarbonate and CO2 have really different isotopic signatures from their carbon-13. One of them sits around 0, one of them sits around -10. And if you look to the right, you are what you eat in terms of a seaweed, you are what you photosynthesize. So we can actually see what kinds of carbon and what the proportions of carbon are that the seaweeds are using based on their isotopic ratio. If you see on the far right, there's a lot of overlap there, but basically if you have a carbon isotope ratio of less than -30, this purple box, you're only really using carbon dioxide with these values. So this is a pretty cool trick, but this isn't the only way we can find out what kinds of carbon seaweeds are using. We can think of isotopes as kind of a long-term, time-integrated metric that can develop over months or a whole season. But there's also a really short-term metric called a pH drift assay. Remember when we said, let's make this really simple, there's a threshold for a pH drift assay if you incubate a seaweed under light in water for, say, 24 hours. If the seaweed is able to drive the pH of the seawater up past 9.0, then it must be using bicarbonate because there's no CO2 in water with a pH greater than 9.0. So what we could do is compare these short-term assay results with long-term isotope assay results, and that's what this plot is doing here. We would expect that if these two metrics agreed with each other, CO2 users would only fall into the lower left quadrant, while bicarbonate users and CO2 too, using both would fall into the upper right quadrant. And that's actually what we see pretty well. These two metrics are doing a pretty good job together. What this means is we can use carbon isotope data to get a pretty good handle on bicarb use in seaweeds. And that's great because the pH drift assay is a pain in the butt to perform. It's difficult, it's time intensive. Not very many people do it out there, but there's a huge literature of carbon isotope data. So I assembled this study meta-analysis from 76 studies that have carbon isotope data from all over the world. So you can see here, all these red dots are sites where we have species that researchers have gone out and collected carbon isotopes for. So thank you all so much for that. So if we zoom in and look at what's happening along the latitudinal gradient, so as we run from, say, the equator, as we go out from the equator towards either of the poles, that's what this number 2 graph on the bottom is showing, it turns out that the isotopic signal of seaweeds is lowering the further away we get from the tropics. And this means that we're seeing more seaweeds that rely on CO2, on carbon dioxide, the further from the equator we get. If we look at number 3 here, we see that actually brown and red algae are driving this trend. These yellow stars here indicate that these are significant trends based on the statistics that I ran. But we don't see this in the green algae, and we don't see it in the light blue here. It represents surf grasses, other marine flowering plants. So really, brown algae, represented in the black here, and the red algae, the red clearly, are what's really driving this latitudinal trend. So, and I don't show you the data here, but I will say that variation within a species is very low. So in their isotope value. So it would seem as though we're seeing different species. There's species turnover as you go over the poles rather than a single species just having a really wide range of isotopic values. We don't see that much in the data set. Some other cool trends that we see, the seaweed isotope values really varying a lot by the oceanic basin membership. So this might have something to do with what the source seawater carbon isotopes look like. So the carbon dioxide and the bicarbonate that the seaweeds are eating, those might have different baseline isotopic ratios based on what ocean they're from. If we look at the data in terms of whether you're a green, brown, red seaweed, or a surf grass, if we look at the evolutionary lineages of these different species, we see really different trends. Here you got the dotted line, means to the left of the dotted line, those species can only use carbon dioxide, and to the right, they can use CO2 and bicarbonate. So the red algae show a really different trend. They have a lot of species that can't use bicarbonate at all. And this may have something to do with a vertical gradient that we saw as well. Intertidal species have increased reliance on bicarbonate compared to subtidal species. And a lot of the subtidal species are actually red algae. So maybe that's what's driving this trend, this altitudinal or vertical trend that we see in the data. So I'm going to wrap this up real quick. Again, bicarb use is non-randomly distributed at a lot of different scales. This might have implications for climate change and how the ocean chemistry is changing with increased CO2 in the atmosphere that's getting sucked right into the ocean, even as we speak now. We're seeing more bicarbonate use in the tropics, so along the equator, and also in areas that have really high annual temperature variance, so areas that exhibit a high degree of environmental variance. Now whether you call yourself a brown, green, red seaweed or a surf grass, that actually matters too and has some implications for your carbon use patterns. And overall, we see, based on our comparison of two different metrics, the short-term and the long-term metric, that isotopes generally predict bicarbonate use pretty well. So thanks for listening, guys. I hope I've convinced you that seaweeds are worth your while. And keep an eye out for some other cool stuff that the Journal of Ecology is up to over the next year, all right? Take care.